Okay, good morning. Welcome back. Um, Meanwhile, has everybody gotten a chance to go and look on REM-CT? And you noticed everybody has the syllabus, hopefully, and you noticed the lectures are on there, and also actually the taped lectures as well as the slides. Everything is there for your perusal. I'm glad you're back, though. Hopefully you'll get a little bit more here than if you just watch. Last time we started on a recap of plant anatomy, and we're going to look at the whole plant, and we started looking at the cell. After this recap, which is Module 1, we'll have another sort of recap, which is Module 2, which focuses on metabolism, energetics, gene expression. Just to refresh on what you know about those things, which are very basic, and then we're going to delve into plant-specific processes in relation to water, and then to nutrients, inorganic nutrients, how they take them up, which ones they need and why. That's the end of Section 1, and then in Section 2, we're going to look at carbon, how they acquire carbon via photosynthesis. And then we're going to look at plants' responses to their environment and their growth and development and the roles of hormones. That's the last section. So this slide you haven't seen, the last one was this one. What we have done so far is we've kind of walked through the endomembrane system. And vacuole was the last compartment we looked at. So what the endomembrane system is kind of an assembly line for the synthesis of proteins and lipids. And it starts in the nucleus, which is where the DNA is, but it's also the place where proteins are synthesized here on the outside of the nucleus. If a gene is expressed, then a messenger RNA is copied from that gene, from that piece of DNA, and that messenger RNA gets translated into proteins on the site of where the ribosomes are, which are these black dots that you see on the outside of the nucleus, which is the start of the ER, which is around the nucleus and contiguous with it. So once you're inside the lumen of the nuclear envelope, you can travel right into the ER. And a lot of proteins that end up in the endomembrane system are born into the ER. So as they are being produced, as the amino acids are assembled on the ribosome, the protein is dumped into the ER. And then it goes on its way in the form of vesicles. These vesicles can pinch off of the ER. They can travel. This can be directed by the cell. There are kind of highways in the cell, which means these vesicles can be moved. There are motor proteins that hold them and move them along these roads. And then they go from the ER to the next compartment of the endomembrane system, which is the Golgi apparatus, which you've seen last time. It looks like a stack of pita breads. And proteins and lipids are synthesized from scratch, you could say, in the ER, and then they travel in vesicles to the Golgi, where they are finished. So additional groups are added onto them, like phosphate groups and sugar groups. And these function sometimes as address labels to tell the cell where this protein or lipid needs to go, or they may be needed for the function of this protein or lipid. It's like sugar groups on proteins or lipids that end up in the plasma membrane may have a function in recognition if they are receptors. So once they are finalized, these proteins and lipids are then shipped again in vesicles to the final destination. And from the Golgi apparatus, that final destination may be the vacuole. And in this cell, I didn't find a good picture in a plant cell, so I got an animal cell here. In this animal cell, the lysosome is the animal equivalent of the vacuole, but as you see, it's a kind of a pitiful vacuole. It's very small. In the plant, the vacuole can be 95% of the cell sometimes. And in part, the vacuole in the plant has the same function as the lysosome in animal cells, in that it recycles, it breaks down molecules. So lysosome or vacuole, that's one compartment where these proteins and lipids may end up from the endomembrane system. Another place where they may end up is the cell membrane. So they may just incorporate in that membrane, and that's their final destination. Or they may go right through the plasma membrane and end up in the cell wall, so the apoplast. And we're going to have a look at the cell wall later in this module. So then they don't get stuck in the membrane, but they go through. And then they may also travel away from the cell to other plant parts if they go into the cell wall. So that is the endomembrane system. There are additional organelles that you haven't seen yet, and they do not get their proteins and lipids from the endomembrane system. They are a little bit independent or semi-autonomous. And those are the ones that we are going to look at next. So these are the last three organelles: the microbodies, also called oxysomes, the mitochondria, and the plastids. You've probably heard of the chloroplast. That is a kind of plastid. Here you see an electron micrograph where you see all three of these organelles in one picture. And the first organelle we're going to have a look at is the peroxisome as an example of an oxysome or a microbody. It's here in this red square. And then you see to the right of it a mitochondrion, and to the left and the bottom of it a chloroplast. So it gives you an idea of the sizes of these organelles. The peroxisome can be recognized under an electron microscope because it often has what looks like a cube inside, a very crystalline structure. And this is because it's full of enzymes that do a particular function. Namely, it takes off hydrogen atoms off of a molecule. And if you want to write down some molecule, any molecule, then people often say R. So R can be any molecule that has hydrogen on it, RH2. And the enzymes in the peroxisome take the hydrogen atoms off of a molecule and give it to oxygen. And then you get hydrogen peroxide. Maybe you know this compound because it's, you can use it to disinfect things or to bleach your hair. So this is a pretty aggressive compound, so it's something that the plant doesn't want to accumulate. It's actually, it causes stress. So the plant then proceeds to break down this hydrogen peroxide immediately into harmless water and oxygen again. So, and the plant uses this to detoxify certain molecules. That's one function. And another function is breakdown of fats. Fatty acid molecules also have a lot of hydrogen on them, and in the breakdown of these fats, these hydrogens have to be taken off. And that also happens in the oxysomes. And in particularly seeds, the breakdown of fat is very important because seeds are often small and they need to travel away from the plant. Far away is good because then you have a bigger chance of finding an unshaded place. So seeds are often small, and the plant wants to use the seed as a even though the seed is small, it wants to put a lot of energy in that seed so that the seedling can grow and has a chance to survive. And fat is the best compact way to store energy. It has the most energy per volume or per mass. So that's why seeds often have fat in them. And when a seed germinates and the seedling has to start growing, then this fat has to be broken down and turned into sugars. That's called gluconeogenesis. So gluco, that's sugar. New, genesis is the creation. So the creation of new sugars using fat as a starting point. That happens a lot in seeds as they germinate. And for that, these oxysomes, a special kind of oxysomes, are important. And they're called glyoxysomes. And they can't do it all alone. That's what you see here on the right. Here you have the lipid body, which is full of these lipids, these fats. And then you have the glyoxysomes working together with the mitochondrion, which you will see in a minute, and the cytosol. And this, you see a series of reactions that together work to break down the fat and turn it into sugar that the seedling can use to start growing. So often organelles will have to work together, and these molecules, these intermediate metabolites, have to be shuttled from one organelle to another across the membranes. So those were the microbodies, or the oxysomes, the first of the three semi-autonomous organelles that import their proteins and lipids from the cytosol, or sometimes they can also make them themselves. Mitochondrion we see here, and we saw it earlier in the electron micrograph that I showed with all the three organelles, and here you see another one, other electron micrograph, and these are the mitochondria. So they kind of look like they have all these vesicles, you could say, in them, but in fact, if you look at this drawing, what it is there's a membrane all around the mitochondrion, and then you have a second membrane inside, which is very folded, and you see that here. And these folds are called cristae. And the purpose of having folds in organelles is to create more membrane surface, so you can have complexes in there, protein complexes that can do something, some function. So proteins that need to be in membranes, and to create more space for these protein complexes, you fold the membrane very much. So that's what you see here in the mitochondrion. So what happens in these mitochondria, the machinery in there is involved in respiration. And respiration means breathing. We respire all the time. And that's because we have a process called respiration, and plants have it too, where sugars are broken down by reacting with oxygen, and they're broken down into CO2 and water. And sugars, as you can imagine, have a lot of energy. CO2 and water don't have much energy. So in this conversion of sugar to CO2 and water, we have a lot of energy being released. And this energy is captured in the form of ATP, which you see here. ATP is kind of like the currency of energy in cells. Like the dollar bill is the currency of money, of something valuable. So if you have something valuable and you want to get something else that's valuable, you often sell first the one thing that you have and then use the money to buy something else. So similarly in the cell, if you have a molecule that has a lot of energy and you want to make another molecule that has a lot of energy, then you can break it down first, the energy-rich molecule. You harvest the energy in the form of ATP, which is mobile, it's a molecule, and then you can use that energy elsewhere to build whatever other molecule you want to make. So most of the energy in the cell is made in the mitochondria, right here. And you will see a lot more about the process of respiration in module 10, I think, or 11. So that's section 2. But basically what happens is hydrogen moves from sugars to oxygen. So then if you take the hydrogen away from sugars, which are basically carbohydrates, or CH2O, if you take the hydrogen away, you are left with CO2. And if you add the hydrogen to oxygen, then you get water, H2O. So, and those hydrogen atoms move in the cell. And as they move across the membrane, and thereby they release energy, and this energy is then harvested in the form of ATP. So you see that here schematically, and you'll see it again later.
I have a quick question.
Yeah.
So is the ATP stored in the cristae? Is that?
The ATP can be anywhere, so it can travel around in the cell from one place to another. So it's not necessarily stored there. So the two membranes of the mitochondrion are called the outer and the inner membrane, and the folds are called cristae. And the space in between the membranes is called the intermembrane space. Makes sense, everything? And mitochondria used to be free-living organisms, and after they were engulfed, eaten by some other cell, some way back in history, the cell recognized, hey, this is a useful organism that I just ate. Maybe you can become my buddy, and you now can serve as my organelle and make energy for me, and the rest is history. So now all organisms, all eukaryotes have these mitochondria. Plastids, the third organelle that are semi-autonomous, a little bit independent. They can make some of their own proteins and lipids, or they import them from the cytosol. Plastids are a group of organelles that include the chloroplasts, which occur in green plant parts. And other plastids are chromoplasts, which often have beautiful yellow or orange or red colors. You find them in bell peppers or in flowers, like sunflowers. There are amyloplasts, which specialize in starch storage, like in potato tubers, for instance. There are leucoplasts, which are just white and kind of nondescript. And then there are proplastids, which are kind of baby plastids that can turn into other chloroplasts. And actually, plastids are pretty flexible. So you probably notice if you have a potato and you leave it in the light, it can turn green. So then an amyloplast may turn into a chloroplast. Or if you put a plant in a cupboard and you close the door for a week and you open it, then it's not green anymore. The plant has turned white or yellowish, and then the chloroplasts have turned into leucoplasts. So they are plastid. Plastids are plastic. They can turn into one another. The plastid is the most complex organelle, and it also used to be a free-living bacterium, a cyanobacterium, blue-green algae, but now it's an organelle which photosynthesizes for plants. So photosynthesis means photo is light, synthesis is production. So it produces something with the help of light. What does it produce? Sugar. So it uses CO2 from the air, combines it with water, and makes sugar, which is amazing that it can do that at room temperature. But it has a pretty complex structure to do all that, and we'll look in more detail in section 2. But here's the anatomy. There's an envelope that consists of two membranes, the outer membrane and the inner membrane. So you see that here. And then once you're through that envelope, you're in the stroma, as it's called. It's kind of like the cytosol of the chloroplast. And then there's a third membrane called the thylakoid membrane, which is very folded. And that tells you there's probably something important going on there. Some protein complex is doing something important. Yes, those are the protein complexes that harvest the light energy. And they store it, again, in the form of ATP, as you see here. Here's a membrane. And again, we have hydrogen being shuttled from one molecule to another. If you look at the reaction of photosynthesis, we have CO2 and water being combined into sugar and oxygen. Looks familiar. It's the reverse reaction from respiration, but it happens in a different organelle. And like I said earlier, if you go from sugar to CO2 and water, this releases lots of energy. And so you can imagine the reverse reaction requires a lot of energy. So that's why light is needed to make that happen, to provide that energy. But light energy first has to be captured before it can combine these molecules into an energy-rich sugar molecule, and that temporarily that light energy is captured again in the form of ATP, the universal currency of the cell. And the mechanism that the chloroplast uses is very similar to what the mitochondrion uses. It can allow these protons, these H+ ions, to first accumulate in one compartment, then flow through a membrane, through a special molecule called ATP synthase. It's a protein complex that can then couple the flow of these protons, these H+, to the synthesis of ATP. And then that ATP is used in that same chloroplast, in the stroma, to make the sugars out of the CO2 in the water. It's basically out of the CO2 by adding the protons to the CO2. So we'll see more about this later, but it's not a coincidence, incidentally, that this process of ATP synthesis in the chloroplast and in the mitochondrion is so similar. Probably it has the same evolutionary origin. And probably photosynthesis was there first. Because photosynthesis produced all the oxygen that was in the atmosphere before respiration could get started, because respiration needs all that oxygen. So that is just an interesting evolutionary thing to think about. I mentioned a couple of times that these organelles, particularly mitochondria and plastids, but probably also peroxisomes, they're semi-autonomous. So they don't depend so much on the rest of the cell. They're a little bit more independent, and that probably reflects that they are fairly recent organelles, relatively speaking. They still have some of their own DNA, which is prokaryotic in nature. I mentioned earlier that eukaryotes have their genome, their DNA, arranged in the form of a linear set of molecules. Prokaryotes, on the other hand, have one circular molecule of DNA. And that's what you still see in these plastids and these mitochondria. They have a circular piece of DNA. And this DNA encodes proteins. About maybe 10 or 15% of the proteins in these organelles are made in the organelle themselves. So that means they have the whole machinery of messenger RNA synthesis, translation, they have their own ribosomes. And this whole machinery also bears resemblance to prokaryotes and not to eukaryotes. So they're still very bacterial molecularly. And there's a term for this. The people who figured this out came with this hypothesis, this endosymbiont theory. A theory in evolution is kind of a hypothesis that's very well supported. So it's kind of everybody agrees that this is probably true. So endosymbiont theory is that these organelles, many of these organelles, definitely mitochondria and chloroplasts, used to be free-living organisms and now they're organelles, and they're still a little bit independent. In addition to doing photosynthesis, plastids have other essential functions for the cells. So it's not just carbon reduction, but also sulfur and nitrogen reduction and assimilation. So going from inorganic nitrogen and sulfur to organic forms, amino acids, the plant depends on the plastids to do that. And photosynthesis is carbon reduction and assimilation. So carbon, sulfur, nitrogen, they're all reduced and assimilated in plastids. And when I'm talking about reduction, I'm talking about a kind of chemical reaction where electrons are added onto molecules. That's reduction process. Reduction and oxidation. If a molecule gives up electrons, it's being oxidized. If a molecule accepts electrons, it's being reduced. So redox reactions are reactions where two molecules interact with each other. One gives electrons to the other. So the one that gives the electrons is being oxidized. The one that gets the electrons is being reduced. So where there's one reducer, there's always an oxidizer at the same time. So it's a redox reaction, reduction, oxidation. And the respiration and photosynthesis that you just saw, they are examples of redox reactions because we have electrons moving, either from sugar to CO2 or the other way around from water to CO2. And they take hydrogen along, H plus protons and electrons. They move together in respiration and photosynthesis. Just a little set the stage. We'll come back to that later. So now you've seen all the organelles in plants. So let's recap. We've seen the nucleus. We've seen the ER. We had rough and smooth ER. We had Golgi apparatus. And we had the vacuole. They're all part of the endomembrane system. They're all organelles because they are membrane enclosed. Other parts of the endomembrane system, you can argue, are the plasma membrane and the apoplast. They also get their proteins and lipids from that origin. And then there were the three semi-autonomous kinds of organelles, which were the oxysomes, or microbodies, the mitochondria, and the plastids. And most of these organelles occur in other eukaryotes as well, but the plastids do not. They are plant-specific. And then the vacuole is also quite different in plants than in other organisms, other eukaryotes, because it's much bigger. And it has this sturdiness function in addition to recycling function, and a big storage function as well. So the vacuole is kind of special in plants as compared to other eukaryotes, and plastids are unique. Another thing that is unique for plants, definitely compared to animals, is the cell wall that is surrounding the cell membrane. It's kind of a wooden box, and it gives the cell sturdiness. Fungi also have a cell wall, but it has a different structure from the cell wall in plants. Animals do not have cell walls. So animal cells are much more sensitive. They break much more easily. Plant cells are protected. So what we're going to do next is look at some of these other structures in plants, including the cell wall, which are not technically organelles because they're not membrane enclosed, but they're interesting enough to have a look. So we're going to have a look at the cell wall. We're going to have a look at the cytoskeleton, which is really what it says. It's a cell skeleton. It's made up of proteins. And then we'll have a quick look at the ribosomes again, which you have already seen. So these structures function in protein synthesis. Those are the ribosomes. Support, which is the cell wall, and the cytoskeleton, also for support for the cell. And then cell-cell communication, which is particularly the cytoskeleton, you could say, and the cell wall. So ribosomes you've seen earlier when we looked at the ER. They were those black dots that can attach to the ER, make a protein, and then let go. They can also make proteins in the cytosol, and those proteins are destined for other destinations. If they are made in the cytosol, they either stay there or they go to the chloroplast, mitochondrion, or oxysome. So these ribosomes that you see here as black dots, they may be synthesizing proteins as we look at them, and if they are bound to the ER, they're definitely making proteins. So what is a ribosome? It consists of two parts, a large subunit and a small subunit, and it consists of two kinds of macromolecules, namely RNA and protein. So ribosomes, they're pretty big. You can see them under a microscope, and this gives you the scale, gives you an idea, 0.5 micrometer. So they're pretty big, relatively speaking, for a molecule. It's a big complex of RNA and protein, and its function is to synthesize proteins out of messenger RNA. It can read the nucleotide sequence of messenger RNA and then translate that into an amino acid sequence in the protein, in the polypeptide, where 3 amino acids encode for three nucleotides encode for 1 amino acid. So that's what ribosomes do. Cytoskeleton, second kind of macromolecular structure that in cells that is not an organelle. Cytoskeleton is indeed sort of a skeleton of the cell. It is made of proteins, and there are 3 different components of these protein fibers. And together they function in support, but also in movement. So support, for instance, the nucleus, is always located in the same place in the cell, where it's anchored in place by the cytoskeleton. So the nucleus doesn't move around freely, it is where it is, and it's anchored in place. It's kind of like nail and hammer. You use the cytoskeleton as a building material to put something in place. Also, if a cell has a certain structure, like a shape, that shape can be maintained by means of the cytoskeleton, so it doesn't move all over the place, but it keeps its shape. And then movement, if you think of vesicles moving, cytoskeleton makes that happen. Sometimes you see under a microscope cytoplasmic streaming. Maybe you've seen that in some aquatic plants under a microscope in freshman bio. That's also caused by the cytoskeleton. That's another example of movement. So molecular movement. These cytoskeleton components can be very flexibly used to make a highway, like a monorail, to move something, and then it can be dismantled equally fast, and then those components can be used to make a new highway. For instance, if a cell divides, then you probably remember from genetics that the chromosomes line up in the center of the cell, and then they're pulled apart by the spindle. And these spindle components are another example of cytoskeleton, and they move the chromosomes. That's an example of movement, cytoskeleton being involved in movement. And this is very temporary because you don't need that spindle there all the time. It would only be in the way. So it's produced temporarily, it does its job to move things, and then it's dismantled again. So there are 3 components that vary in the thickness of the protein fibers, and they are called the microtubules, the microfilaments, and the intermediate filaments. And the microfilaments are the thinnest, the microtubules are the thickest, and the intermediate filaments are, as the name implies, intermediate in size. And here you see on this picture a picture of A, the microtubules, and a picture of B, the microfilaments. So you see A is much bigger, much thicker in diameter than B, and you can then imagine that the intermediates are somewhere in the middle. The microtubules are the only ones that are actually hollow, and you see that space here, and the other 2 are solid. And all 3 are made of proteins, but different proteins. The microtubules are made of tubulin. That's a kind of protein. And you see one, to give you an idea, one polypeptide is kind of this halter-shaped object here. That is like 1 unit of tubulin. It has 2 polypeptides, the alpha and the beta chain. So that together forms 1 unit, and then a whole bunch of these same units are coupled together to form this hollow structure. So you can imagine how many protein molecules are in one of these fibers, because each is one. So tubulin is the kind of unit, subunit, that is used to make these microtubules. Actin is the kind of protein that microfilaments are made up of, and the intermediate filaments are made of keratin. And keratin you probably know because your nails and your hair are made of keratin, so it's the same protein that is used in the cytoskeleton. So here we see a little bit more about these guys. Microtubules, they were the thickest. They are straight, they're hollow, and they're very rigid, so they're not flexible. And they are used as monorails, so like a superhighway, to move things in the cell. An example of movement you see here at the bottom, where you have a motor protein, which is this red thing, kind of with legs, and it can actually move those legs. And there are some cool animations you can find on the web. If you look at Cells Alive, I think it's called, then you can kind of see these legs move. And then the cargo, they're kind of holding a cargo on their head. And that's how those cargo things are moved. And movement always needs energy. Nothing is for free. So guess which molecule is providing this energy? ATP, our friend ATP. So that's one place where ATP is needed after it's made in the mitochondria to move things, like to move vesicles or organelles. And the movement itself is done by motor proteins, and they use these microtubules as the highways to move along. Another way in which these motor proteins can make things move is this way you see here on the top. Two microtubules can actually slide along one another when these motor proteins that are attached to one of the microtubules walk along the other microtubule. And this is, for instance, how in a spindle, where chromosomes are pulled apart, the chromosomes are actually dragged to the two ends of the cell. We have these microtubules which are aligned in parallel along one another. They start to slide along one another, and then the one that's sliding down is holding the chromosome. So that's how the chromosome is going down. So two microtubules can slide along one another. Again, motor proteins use energy in the form of ATP to make that happen. An example of movement mediated by the cytoskeleton. So movement in cells is not random. There's a plan behind it. Microtubules. Here you see a spindle. What they did in this example is they used fluorescent labels to label different macromolecules in the cell. And the DNA was labeled here in blue and the microtubules in green or yellow. And so this way you can actually see, you can arrest them in different stages of cell division and then stain these molecules, and then you can kind of see what happens to them. So that's another example of movement mediated by the cytoskeleton, by microtubules, pulling apart chromosomes during cell division. Going from the microtubules, we now go to the microfilaments, which are the thinnest. Because they are so thin, they are particularly suitable for supporting very thin structures. And here you see an example from the animal world. This is not a plant cell. This is an intestinal cell, which has a very enlarged surface area so that that intestinal cell can easily take up lots of nutrients. And these protrusions are called microvilli. And they're so thin that they can only be supported by the microfilaments. They're thin enough to support that structure. So similarly, in plant cells, if there's a very fine structure, then it likely will be supported by the microfilaments. Microfilaments are intermediate in permanence between the microtubules and the intermediate filaments, where the microtubules are the most ephemeral, they're the most short-lived, very temporary. The intermediate filaments are the most permanent, so once they are in place, they never leave, and the microfilaments are intermediate. They're also intermediate in terms of rigidity, so they're a little bit flexible, but not as flexible as the intermediate filaments, and the microtubules are not flexible at all. So they each, as building materials, have their advantages. An example of a process in plant cells that is mediated by microfilaments is the cytoplasmic streaming that I was talking about. So that's a kind of movement that you can see under a light microscope. You can see things, the cytosol with some small organelles just moving around a vacuole. So a little bit of movement sometimes, but also just support is the function of the microfilaments made of actin. And actin may also ring a bell because it's used in animal cells for muscle contraction. Myosin and actin, where myosin is a motor molecule like the red legs that you just saw in the previous picture. And the actin is the cytoskeleton component that gets moved around to contract and expand. So intermediate filaments, they are the most permanent, they are the most flexible, they are purely meant for support, putting things in place, so not for movement. Kind of really like a skeleton of the cell. And here at the bottom you see all three of them one more time. So the microtubules, they're hollow, they're the thickest, they're very rigid, very stiff. Then the actin microfilaments, they're the thinnest, they're a little bit flexible. And then on the right, the intermediate filaments, they're intermediate in thickness, they're very flexible, they're very permanent. Intermediate filaments, very permanent. Microtubules, very temporary. Actin, the microfilaments, intermediate. So that was the cytoskeleton, ribosomes, cytoskeleton. Lastly, the cell wall. It's another example of a structure that is pretty big. You can see it under a microscope. It's not an organelle because it's not enclosed by a membrane. It is unique for plants, definitely in this chemical configuration. And it has as a function support for the plant, for the cell and for the plant as a whole. The cell wall is made up of basically three components, but I've shown here only cellulose. But what it starts with, if a new cell wall is being formed, is vesicles containing pectin, which is very sticky. People use it to make jam. It's very sticky. These vesicles of pectin fuse with one another in the middle of the cell, and they form an initial middle lamella, as it's called. And then cellulose fibers are deposited against this middle lamella from both sides. And that forms the primary cell wall, as it's called. So primary wall, shown here, is this layer. And the middle lamella is shown as this dark layer. That was the original initial separation between two daughter cells after division. The primary cell wall is made of cellulose. And you know cellulose, for instance, by cotton. Cotton is made of cellulose. It's a carbohydrate. It's made of glucose. It's insoluble. And it's very strong. It's made of, as you can see with cotton, that's pretty tough if you try to pull cotton apart. So that is the primary wall. While the primary wall is there, the cell can still grow. It can expand. And then once the cell is fully grown, a secondary cell wall is deposited against it. And that's what you see here. So we have three layers of secondary wall, 1, 2, 3. And these are depositions that contain a lot of lignin. It's a pretty complex chemically bunch of molecules, but it has a lot of lignin, which has ring structures in them. And this is even stronger than cellulose. And you can realize that when you think of paper and wood, that is lignin. So that is pretty strong stuff. And as you will see in the next topic, which is water relations, module 3 and 4, you will realize, you get an appreciation for how strong the cell wall is, because the pressure, the turgor pressure that builds up in plant cells is actually higher than in your bike tire or in your car tire. That's pretty amazing. And those cell walls don't break. And that's what gives the plant its sturdiness. That's the difference between turgid and wilted. That's the cell wall makes that possible, to blow those cells up with water. Nowadays, there's a lot of interest in the cell wall because people want to create biofuels from cellulose. So there's a lot of research going on in this topic. This gives an idea of how the cellulose fibers are deposited as they against the middle lamella from the plasma membrane. And you see there are these pores in the plasma membrane where the cellulose is growing, you could say, like a hair out of a skin. And the sugar units are added onto it in this protein complex, and then the cellulose fiber is deposited in a specific orientation, and the plant uses as kind of to get maybe as an orientation, it uses microtubules, which you see here in yellow, and then it puts it along the length of the microtubules. And it's important for the cell that these cellulose fibers are all arranged in parallel, because that makes it possible for the cell to keep expanding. Because if you would put those cellulose fibers crisscross, you couldn't pull them apart. They would all be too much entangled. But as long as they are still parallel, the cell can still keep growing. And then when the cell is done growing, it can actually make cross connections between these parallel fibers. And that links the cell wall so it doesn't grow anymore. And this is actually reversible. So if a cell decides it needs to start growing again, it can release those bonds between the cellulose fibers. Cell can grow. Turgor pressure can push it so that it grows. And then those links can be reestablished. And we'll see that when we look at plant responses to light or gravity, because then one side of the cell makes that happen, the other, not the cell. One side of the plant makes that happen, allows itself to grow, and the other side of the plant does not, depending on where the light comes from. And that's how one part of the plant grows, and the plant grows in a certain orientation. So that's how cellulose fibers are produced. And one thing to reiterate, which I mentioned before, the cell wall forms kind of a box around every cell, but there are holes in this wooden box, and these allow for cytosolic connections between neighboring cells. And those are called plasmodesmata, and that is a Greek word. Desma is the singular, plasmodesma, and plural of that is desmata. So it's just the way the Greek do plural. So these plasmodesmata are connections of the cytosol between two neighboring plant cells, and they allow for molecular traffic. And even RNA molecules can travel through these holes, these plasmodesmata. And viruses can travel through the plasmodesmata. There is actually some regulation, so there are some gatekeepers there which control who goes through. But I guess the viruses talk them into letting them through. They hijack the system. So cytoplasmic connections between cells are called plasmodesmata. And these plasmodesmata allow symplastic transport. Remember, the symplast is the space in an organ that is within the collective plasma membrane, which is connected by means of these plasmodesmata. So everything within the blue line is the symplast, and that allows for symplastic transport of molecules. There's also apoplastic transport, and that you see here as the pink line. And you will see these lines again when we look at how nutrients move, how water moves. So this is pretty basic concepts that we will see again. If the plant has no choice, then it will use transmembrane transport. Energetically, it costs more to move things across membranes all the time. So if it doesn't have to, it will probably choose to go either symplastic or apoplastic, because then there are no membranes to cross. That's much easier. And you might not realize it if you think of a wooden box, but the cell wall is actually full of holes, so molecules are small enough to freely move through the cell wall. Cell wall is not really a barrier. Unless you're an organism like a fungus trying to grow, then the cell wall can be a barrier, but not really for molecules. So that was basically everything about cells and cellular structures. Can I help answer any questions, or does anybody want to add anything before we move on? Yeah, I see two questions. The microfilaments, or do you mean the cellulose fibers? The microtubules, the microfilaments, and the intermediate filaments, they are part of the cytoskeleton, and they have two basic functions: support and movement. So examples that you saw was vesicles moving, organelles moving, chromosomes moving, cytoplasmic streaming, that's movement, and support is just cell structure.
So, two sides very sure that they were parallel, and then they go crisscross, but then they're still the same thing, you're just showing how they're...
The parallel and the crisscross that I was talking about, that was cellulose fibers, carbohydrate fibers that are part of the cell wall. So they give the cell sturdiness from the outside. So they're part of the box around the cell. And I was talking about crisscross versus parallel, saying, look how parallel they are, and that has a function, because if they're parallel, you can kind of pull them apart easily, because the cell is still growing at that stage. If they would be crisscross, then you wouldn't be able to still grow as a cell. And then I said also that they're parallel, they could be teased apart, however, they can be cross-linked at will. So a cell, while the cell is still in that stage, it can cross-link them or remove the cross-links. That's regulated by hormones, and that can explain some of the growth responses of plants towards light or towards gravity, as you will see later. Does it make more sense? Okay. Other questions? Oh, if, for instance, if there are no cytosolic connections, in some cases where plants need to have more control, if, for instance, into the plant from the soil, there are no cytosolic connections there, otherwise the plant would just leak all the time. So there are no holes in the outside layer of the plant. So then anything going into the plant has to pass a membrane. And that gives the plant the possibility to control the trafficking. Similarly, if a plant is going to transport molecules from one organ to another organ, then there will be no plasmodesmata all the way. Then there are membrane barriers, which gives the plant control. It can store something, accumulate something in one place without it leaking out. So there it has to pass membranes. And also, of course, within cells, from one compartment to another, like from the cytosol to the chloroplast, the membrane transport is needed, because there are no holes to get into organelles. So there, again, membrane transport is needed. Yeah?
You said the nucleus is permanently fixed, but it's attached with like the temporary microtubules.
It's attached with the intermediate filaments, which are the most permanent part of the cytoskeleton. The nucleus is attached with these cytoskeleton components just somewhere in the cell on the edge against the cell plasma membrane. Yeah. Okay, great. Well, I'm glad you had questions. That does mean that we're going to stop now, so we'll take it from here next time.